Hello everyone. Today I'll be talking about PlasBin-flow, which is a flow-based MILP algorithm for binning plasmid contigs. First, we need to talk about why we are interested in plasmid. And the main reason is their role in spreading antimicrobial resistance. We then move on to discussing plasmid binning, which is grouping contigs into plasmid bins. In our method, we use information provided by the assembly graph, which we'll discuss in detail later. And we also use network flows and combinatorial optimization techniques. So our tool is called PlasBin-flow, and it is available at this address. Plasmids are short circular molecules of DNA that are separate from the chromosome, and they can occur in multiple copies in the bacterial cell. They usually carry genes that are beneficial for their host bacterium, and mainly some of these genes are AMR genes, which give the host resistance against drugs. Plasmids mediate the spread of these genes in bacteria through horizontal gene transfer. Our input consists of whole genome sequencing data. In particular, we focus on using short read whole genome sequences, and the sequence reads assemble short contigs. In plasmid binning, we try to form a set of contigs that represent a single plasmid. And our job is to compute these plasmid bins from the pool of assembled contigs. In addition to the assembled contigs, some assemblers also provide us the assembly graph. The vertices of this graph are the contigs, while the edges represent potential connections between the contigs. This proximity information that is encoded in the assembly graph can be used for plasmid identification. And it's especially important for correctly binning short contigs. So on the right, we have an example of an assembly graph for an E. coli sample. This was assembled using unicycler. And you can see this is a pretty dense graph with over 200 vertices and edges. The plasmids in the graph are connected components that have been circled in the red and green, while the rest forms the chromosomal contigs. Quite a lot of plasmid binning approaches have been proposed till now. So roughly we can characterize them as de novo and reference based. De novo approaches use plasmid features as well as some features from the assembly graph. So for example, plasmid space and recycler are such methods. Reference-based methods rely on a reference database. So they map contigs onto genes or plasmids in a reference database. MobRecon is one such approach. Hybrid approaches combine good properties of the previous two approaches. So highest G plus plasbin and the current method that we want to that we'll be discussing, PlasBin-flow, form hybrid approaches. What does an ideal plasmid bin look like to us? It should ideally form a walk in the assembly graph. The contigs should have uniform read coverage, and the bin also should allow contig multiplicity. The GC content of contig should be similar across the bin, and the contig should contain genes that are typically found in plasmids. So here on the right, we have an example assembly graph, wherein you see the contigs in green have a high gene density, which is the GD. They have their GC content falling in in a small region, while the ones in blue have low gene density, and their GC content is also disparate from the green ones. PlasBin-flow is, as we discussed, a hybrid approach. It uses plasmid features in the assembly graph, as well as using a database of plasmid genes. It is an optimization-based approach. It tries to maximize an objective function that is defined in terms of the plasmid features that we discussed previously, particularly the read coverage, plasmid gene density, and GC content similarity. Most importantly, it uses the context of the assembly graph to define its components in order to identify the plasmid bins. PlasBin-flow works in an iterative manner. In each iteration, it extracts one plasmid bin, that is the optimal plasmid bin for that iteration, as a set of contigs. It then removes the plasmid bin from the assembly graph, and the assembly graph is then updated to reflect this. The process continues till no further plasmid bins can be extracted from the assembly graph. So on the right, we have an example where we start with the entire assembly graph. Then we have one iteration of the method which identifies the plasmid bin in green. We then update the assembly graph to remove contigs that can be removed from that plasmid. And then we use the remaining assembly graph for the next iteration, where we then continue with the yellow plasmid bin. And this process continues till we don't have any more bins to remove. So we use network flows to guide what an ideal bin would look like for PlasBin-flow. So we add external auxiliary vertices, which is the source and sink vertices S and T. And then we send a flow through S, which comes out to T. And this flow acts as a proxy for the copy number of the plasmid bin. The flow also ensures that we have uniform read coverage throughout the plasmid, and we implement a constraint that indicates that flow into a contig also equals flow out of it. The capacity of an edge in this bin between contigs is defined in terms of the read coverage of the contigs. So here on the right, we have a flow from S to T of value 2. And you can see that we also can define a flow that allows contigs as well as edges to occur multiple times throughout the course of the plasmid. So next we'll look at the criteria for finding the best plasmid bin. We design our algorithm as an optimization-based problem, so which involves an optimization function. So our optimization function consists of 3 terms. The first one refers to the flow, which is a proxy for the overall copy number of the plasmid bin. Next, we have the penalty for deviating from the GC content of the bin. So minimizing this term also minimizes the overall variation between the GC content of chosen contigs. Next, we have the plasmid gene density term. So maximizing this term encourages the algorithm to choose contigs that have a high density of plasmid genes. So we formulated our method as a mixed integer linear programming problem, and we solved it using the Gurobi optimization solver. We conducted experiments on a real data set with 133 bacterial genomes, and that consisted of 377 plasmid. This data set was obtained from the one that was compiled by Robertson and Nash in 2018. So we split the data set into a reference and test set. The reference set consisted of 67 samples with 230 plasmids, released before 19th December 2015. And the use of this reference data set was to mainly determine the gene density of contigs in the test data set. And it also helped us set the parameters that are required to run PlasBin-flow. The test dataset then contained the remaining 66 samples of varying species of bacteria and 147 plasmids. We compared PlasBin-flow against various methods, various plasmid binning methods, and we used the mainly two evaluation metric to evaluate our performance. So the first consisted of weighted or base pair level statistics, which used the lengths of individual contigs uh, to weight the statistics. And the second was the unweighted or contig level statistics, which only considered the number of individual contigs that have been correctly predicted. Then, using these two criterion, we defined the precision, which involved assigning one true plasmid to each predicted plasmid bin, and that true plasmid would maximize the correctly predicted length or number of contigs from that bin. Recall, on the other hand, involved assigning one predicted bin to each true plasmid and then maximizing and choosing the bin that maximizes the correctly predicted length or number of contigs from that plasmid. So based on these definitions of the evaluation criteria, we have the following results. So when it comes to weighted or base pair level statistics, we can see that PlasBin-flow showed decent precision and recall, but it was second best to MobRecon. However, it definitely showed improvement over its predecessors, PlasBin and HIAS. Next, we look at the unweighted statistics, and PlasBin-flow definitely showed good precision here. And all the tools pretty much showed low recall. However, PlasBin-flow had the best overall F1 score. Looking at the evaluations, we can see that PlasBin-flow consistently performs well when it comes to precision, but there is definite room for improvement in recall statistics. The main reason behind this is that an actual plasmid might be split into multiple bins that have been output by PlasBin-flow. And this occurs when groups of contigs that ideally should belong to the same plasmid are connected with contigs that have typically low plasmid gene densities, which means PlasBin-flow does not have an incentive to include these two bins in the same solution. This can be addressed by designing appropriate post-processing algorithms, or we can also try to improve the objective function of the MILP itself. So in conclusion, we have a new plasmid binning tool called PlasBin-flow. It's a hybrid approach that uses information from assembly graphs, and it's based on a well-defined optimization problem rather than a heuristic. Important features of our approach include that it's a flow-based formulation that ensures uniform read coverage in a plasmid bin, even when the plasmids contain repeated contigs. It also uses GC content penalties, which discourages the mixing of two different bins, which is typical in dense graphs. We showed that PlasBin-flow outperforms other tools in contig level statistics. This happens because it's useful in particular in binning short contigs, which are usually difficult to bin correctly.